Gene Prediction Part 2. In this part we're going to look in some detail at the ab initio methods, and then in Part 3 we're going to look at the similarity methods. In prokaryotes, what we can do is known as open reading frame or ORF scanning, and this is a very simple procedure. All we need to do is search the DNA for start and stop codons, which are in frame with one another. We then can search for promoters, which is admittedly a bit more difficult, and then we can take the fact that most genes are at least 50 codons long, so 50 amino acids long, and select some sort of cutoff, often 100 amino acids or 100 codons, as being the shortest gene length. In eukaryotes, things are much more complicated. So if we start with a similar sort of scenario and we search the DNA sequence for start and stop codons, and then we can assume that those are open reading frames, we can then search for promoters as we did before and make the same sort of rule that most genes are at least 50 codons long. So we would throw away that second potential open reading frame. However, if in reality there are intron-exon splice sites at the indicated positions, then the real gene structure looks like this, and the open reading frame and the coding sequence ends up looking like this. How do we do this with ab initio methods? Well, one of the first things we can look at is codon usage. Now, the genetic code has redundancy in it. So if we take, for example, leucine, there are 6 different codons coding for leucine. Valine here, there are 4, and so on. Now, it's a universal feature of all species that there are preferences for which particular codon is used. So we can differentiate coding and non-coding regions by looking for the frequencies of the codons, and regions that are using more common codons are likely to be the real coding regions. So taking this example of valine, then, there were 4 codons for valine, and human exons use GTG 4 times more frequently than they use GTA. So real exons will be expected to show this bias. Random triplet series found in introns should not. Now, this is a complete human codon usage table, and I'm going to zoom in just on the glycine example. So again, 4 different codons for glycine. This is the codon frequency per 1,000, and this is the relative frequency of these 4 synonymous codons. So what we can do then is take a piece of sequence, and we can create some sort of gene finder that makes use of these coding statistics in order to identify the exons and introns. The fact that it's ab initio means that it only uses this intrinsic sequence information. It identifies the coding exons, it integrates these coding statistics, and essentially calculates the likelihood that a particular triplet is in a coding region. The simplest way of applying this sort of information is to use a moving window. So what we see here is a piece of sequence divided into codons and the relative frequency of each codon below that. What we can do then is create a window. In this case I'm using 3 codons, but in reality you'd probably use a wider window than that, and you calculate the average of those scores and plot a point on the graph. You then slide along and do the same thing, and you keep doing that throughout the sequence until you have a plot that looks like this. Now if we do that with a real gene, in this case the beta globin gene, what we see is a graph which very nicely has 3 peaks in it, and here I've marked the regions that are actually exons. So you can see these 3 peaks correlate pretty well with where the exons are, but the boundaries aren't quite right. The other way that we can try to make use of this information is to use some sort of machine learning method. And as we discussed before, that's a general class of computer software which is able to learn from examples and then able to make predictions. So we train it using examples of real data, and the method will learn features of these real examples, and you can apply the trained system then to make predictions. There are many such methods, including neural networks, support vector machines, hidden Markov models, decision trees, naive Bayesian classifiers, and many more. So, in the case of open reading frame scanning, what we could look for here is the intron-exon boundaries to try to get those much better. They do have distinctive sequence features. For example, there is an AG followed by a GT, and the splice site is between those. But only the GT is invariant. The AG is a consensus. At the downstream boundary, it's more variable than that. Basically, we have a string of PYs, meaning T or C, and then an N for anything, then C, A, G, usually. And just trying to do that with looking for those sorts of patterns is very difficult to incorporate into gene prediction algorithms. So these sort of machine learning approaches really help solve that problem. And this was first done back in 1997. So upstream regulatory sequences are also important. This will help us locate where a gene begins. And again, they have distinct sequence features and they're recognition signals for DNA binding proteins. However, these regulatory sequences are very variable. We'll look more at that in the transcription factor binding site prediction lecture. Again, these can be difficult to incorporate into gene prediction algorithms, and again we can use machine learning, particularly for finding things like promoters. And here are a sample of different programs for looking for promoters which use this sort of approach. The best methods, however, for doing the analysis of a genome in order to find the coding regions are based generally on hidden Markov models. So these create a sort of statistical framework and are able to incorporate things like the codon usage, the intron-exon boundaries, and the regulatory sequences. Now, again, there are many methods available. Here's a selection of some of the kind of classic approaches to doing this. And Genscan, dating originally from 1997, is still one of the preferred approaches for doing this. So Genscan identifies the complete exon-intron structures of genes in genomic DNA. It's able to predict multiple genes, partial genes, complete genes, and so on. It uses a hidden Markov model to model the gene structure and actually has separate HMMs for the exons, the introns, and the intergenic regions. It also has different parameters to cope with regions having different GC content. So what we can do with Genscan is take a long stretch of DNA like this and get a prediction out that looks like this. So this is telling us where the initial, the internal and the terminal exons are, as well as where the poly A attachment site is. And it gives us a p-value to say how confident it is in that prediction. Now, this is a probability that the exon's correct, so it's not like a normal p-value where you're looking for a value less than 0.05 for significance. This means high numbers here are good. So a p greater than 0.99 is almost always exactly correct. A p between 0.5 and 0.99 is correct the vast majority of the time, and if you get a value below 0.5, then the prediction's not reliable. In this particular case then, we have 4 of these 5 segments identified very reliably, and one of them somewhat less so. And the prediction looks like this. So here we have an initial exon, we have internal exons, and we have a terminal exon here. And this really emphasizes how the majority of a gene is actually introns. The exons are quite small regions. So, the classic view is that we have a series of exons interrupted by short introns. In fact, it's the other way around. It's the introns that are much bigger in general than the exons. And Genscan can give us a predicted peptide sequence as well. Now, how well do these things perform? Before we can discuss that, we need to discuss how we measure accuracy. So we have, at the nucleotide level, 4 different possibilities. We can predict whether an individual nucleotide is in an intron or an exon. So here we're taking exons as positive and introns as negative. So we can have in this row the reality and in this row the prediction. So for each nucleotide, if we're predicting it's not an exon and the reality is that it's not an exon, then we have a true negative. If we predict it's not an exon but in reality it is, then we have a false negative. Where the prediction coincides with reality and we're predicting that it is an exon and indeed it is, we have a true positive. Where we predict it's an exon but it's not, we have a false positive. And here again we have coincidence of true negatives where we have prediction that it's not an exon, and indeed that's correct. Now, given those 4 different parameters, true negatives, false negatives, true positives, false positives, we can calculate various different statistics. And the first of those is the sensitivity. And this is the number of true positives over the true positives plus false negatives. In other words, the number of positions correctly predicted as being an exon divided by the number of actual exon positions. Now, in the gene prediction world, there's been a somewhat unfortunate error in that people refer to specificity as the number of true positives over the true positives plus false positives. In other words, the number of correctly predicted positions that are predicted as exons divided by the total number of positions predicted as exons. Now, in the machine learning world, where we often use this sort of statistic, this is not called specificity. Specificity refers to something else. It's actually true negatives over true negatives plus false positives. So it's kind of the sensitivity for the negative state instead of the sensitivity for the positive state. What this really is called is PPV, or positive predictive value. However, you will see it referred to as specificity in the exon intron prediction literature. Another statistic that's widely used is the approximate correlation, and this is a rather complex formula but takes into account true positives, true negatives, false positives and false negatives, as does the Matthews correlation coefficient, which for most prediction purposes is probably the best choice of metric. But again, approximate correlation coefficient tends to be used more frequently in this particular gene prediction world. Now we can also calculate accuracy at the exon level. So again, we've got reality along here, exons shown in dark blue, and we've got predictions along here shown in the pale blue. So what we have here is what we can call actual exons, where we have a prediction that goes with them, and missing exons, where we have no prediction that goes with them. So when we have a correct prediction, we have a true positive. So we can calculate numbers of actual exons, missing exons, true positive predictions, and false positive predictions, where we've made a prediction, but in fact there isn't an exon there. Now, in order to do this sort of thing, we need to enforce some sort of minimum overlap, and that might be 50% or 70% of an actual exon overlapping with a positive prediction. So from these we can calculate a sensitivity. So this is the exon level sensitivity, which is the number of true positives divided by the actual exons plus the missing exons. Again, we have an exon level specificity, actually in technical terms a positive predictive value, which is the number of true positive exons divided by the true positive exons plus the false positive exons. We can also calculate some other statistics. We can calculate a missing exon ratio, which is the number of missing exons divided by the actual exons plus missing exons, and a wrong exons frequency, which is the false positives divided by the true positives plus the false positives. So let's have a look at some of the methods that are used for gene prediction and how well they perform. So these are predictive methods used specifically on mammalian sequences, and I've highlighted at the bottom the two that perform best. So you can see we're getting sensitivity and specificity of around 0.9, and the approximate correlation value also approaching 0.9. At the exon level, some of these do rather worse. Obviously you want wrong exons and missing exons to be as low as possible. You want these to be as high as possible. So in general, ab initio gene prediction is good at predicting coding nucleotides. It gets 90% of them correct. It's moderate at finding the exon boundaries, 70 to 75% correct per exon, less for the initial and the final exons, and quite poor at predicting the overall complete gene structure. Less than 50% of predicted genes correspond exactly to the actual genes. The difficulty is really in finding these intron-exon boundaries, and we can actually improve the accuracy by combining different methods. So different methods often predict different elements of an actual gene to high quality, and they can then complement each other in yielding a better prediction. Of course, all of this is very much easier for prokaryotes because we don't have the introns, and therefore no problems with intron-exon boundaries. Now, we can combine ab initio predictions in different ways. So this is taking an example that was done in 2002 using Genscan and HMM-Gene. Now, we can take the predictions and we can OR them together. In other words, we return regions as being an exon if either of the programs predicts it as an exon. So this is prediction 1 from, let's say, Genscan, and this is prediction 2 from HMM-Gene, and if either of them predicts it, then we get a union of the two predictions. This gives us more sensitivity, but it's less specific, so we tend to over-predict. Alternatively, we can AND the predictions together. So what we're doing here is saying if both predictors predict the region is in an exon, then we return that as a prediction. So this is going to be less sensitive but more specific, and we're going to tend to underpredict. Another way of combining them is what's known as exon union intersection. So what we do here is we start with the ANDed prediction, so the less sensitive but more specific one, and then we extend out from this while either of the predictors has a high confidence value, so some sort of significant threshold. And this sort of approach has been used quite successfully to improve the performance metrics. So here Genscan had an approximate correlation of 0.91, as did HMM-Gene, but combining them using this EUI method gave a value of 0.93. And similarly at the exon level, we got values of 0.7 for Genscan, 0.76 for HMM-Gene, and combining them with EUI we get 0.8. So summarising then the ab initio prediction, for prokaryotic genomes it works relatively well. The non-coding component is small, there are no introns, it's really quite straightforward to produce a reliable prediction. For eukaryotic genomes it's less effective. We have larger non-coding regions between the genes which can often confuse us because we may have pseudogenes that look like genes but actually aren't, and also because of the presence of introns. We need to get the intron-exon boundaries predicted correctly, and we also need to find these upstream regulatory sequences to be sure that what we're looking at is a real exon. So we're going to take another break at that point, and then when the third part we're going to look at the similarity methods for prediction.